So today I'm going to talk to you about the role of the microglial inflammasome in developmental white matter injury. So two of the most common contributors to perinatal white matter injury are an infection-induced inflammation and a hypoxic event in utero, and these can lead to preterm birth. So these events also influence the oligodendrocyte precursor cells, preventing them from differentiating into oligodendrocytes. And this is important because oligodendrocytes are essential for wrapping the nerves of the brain in a lipid membrane called myelin. And this is essential for the protection of the axon and facilitates impulse conduction. So in perinatal brain injury, we have a block in this differentiation and a lack of myelin, which is termed hypomyelination. So microglia are the resident macrophages of the brain, and they're known to also be dysregulated with these types of injury, but we're trying to look at the mechanisms of how microglial dysfunction is linked to hypomyelination. And we think that this is through the inflammasome. So the inflammasome is a complex made up of caspase-1 and NLRP3, and when activated, it produces and releases mature IL-1 beta. So in order to look at the inflammasome, we developed this ex vivo organotypic slice culture model, whereby we take the brains of P0 to P3 mice, and we take 4 brain sections and grow them in culture. And over 21 days in vitro, they myelinate normally, and we're able to induce an injury at 7 days in vitro by administering LPS, which is a bacterial peptide, and putting the slices in a hypoxic chamber for 3 days. And when we do this, we see an increase in caspase-1, which is one of the inflammasome proteins, shown here in green, and this is expressed mostly in the CD68 positive microglia, shown here in pink. So this suggests we have inflammasome activation in microglia, and we can show this with Western blotting of the brain explants, where we can see an increase in pro and mature IL-1 beta with LPS hypoxia treatment compared to control. If we look at the myelin, we can see that in control slices that are untreated, they myelinate really well. So myelin is stained here with MBP in pink, which is part of one of the myelin proteins, and neurofilament in green just shows us the axons. And with LPS and hypoxia, we have a significant decrease in the amount of MBP staining that's visible. So to be sure that it's the inflammasome which is influencing the myelination, we can block the inflammasome. So we have gadolinium chloride, which blocks formation, AC-YVAD, which blocks caspase-1, and MCC950, which blocks and inhibits NLRP3. So I've just shown an image here from the gadolinium chloride treatment, and you can see that by inhibiting the inflammasome, we have rescued myelination in these slices by an increase in MBP. So to determine how microglial inflammasome activation inhibits myelination, we looked at the cerebrospinal fluid of preterm versus term infants. So preterm infants are at a higher risk of hypomyelination in development. And also we can see that they have an increased amount of IL-1 beta in the spinal fluid. So the preterm babies are shown here in pink compared to the term in black. And also we can see that there's an increase in follistatin at the same time as the IL-1 beta. So together, these two proteins suggest that they're both involved in injury response. And previous literature has shown that IL-1 beta is sufficient to encourage microglia to produce follistatin. And if we look in our slice culture model, we can actually see that there is an increase in the production of follistatin with our LPS hypoxia injury compared to control. And the interesting thing about follistatin is that it specifically binds to activin A and prevents activin A from binding its receptors and subsequent downstream signaling. And we've previously published a paper showing that activin A signaling is required for developmental myelin. So if we manipulate follistatin in our slice culture model, so here is just an injury with isotype IgG, and we have very poor myelination. And if we use an anti-follistatin neutralizing antibody that will take away the follistatin, we get an increase again in the amount of myelin. So that's showing that we can rescue that effect. So to summarize, we've shown through the development and manipulation of an ex vivo model that the microglial inflammasome activation is sufficient to cause the hypomyelination, and this is due to the production of follistatin, which has interfered with activin A signaling. So I would say come to my poster, but the session was yesterday. The posters are on the website, so I'm number 75, and please feel free to email me with any questions. Thank you.
Thank you, Rebecca. That's really interesting. Is there any question you can ask you can use the link?
Yeah.
But I cannot see any question at the moment. So I can ask you, do you want to expand a bit what could be the targets of future treatments? Where do you see this going next?
Yeah, so at the minute there aren't any drugs which will target follistatin, but there are drugs that are approved for targeting the inflammasome, but they just haven't been tested in a myelination context. So we don't know yet, but that would be interesting because they would be repurposed from other treatments. So for example, there's anakinra, which is for rheumatoid arthritis, which inhibits the inflammasome. So there's definitely potential there.
To use it and see what happens, yeah.
Yeah, yeah.
Congratulations, really good work. Thank you. Thank you.